Monday here. Okay, I'm sure many of you having the Monday blues. Okay, first of all, are you able to hear me? Students, please give me feedback on the comment. All right, are you able to hear me? Is my mic okay? So good morning, Joanne. Okay, I see Joanne is here. Okay, what about other students? Are you here as well? Okay, today I have a interesting topic for you. So this is actually easy to do. Actually, the questions are easy. As long as you know how to use the formula, you should be able to answer the questions, and you should be able to find the answers quite easily. Okay, so it's not a difficult topic, and it's very interesting to learn about it. Okay, good morning, everyone. All right, Yi-Xin. Okay, yes, Yun-Chi, good morning. Wei-Shan, Shino, Wei-Shan, Pei-Shan, Sophia. Okay, good. Let's go straight to today. All right. Now today I'm going to touch about population ecology. Okay. So you see these two words. You have learned what is population. Let me share screen. All right. Now, so I think you can see these. I'm looking at it right now. Okay. Let's go. Right. I will do it as a reference, but I have my own notes. I will discuss using my own notes. Okay. So now here you have seen these two words. You have seen population. Population is what's the meaning of population? Population is referring to the group of organisms which are the same species, okay, that stay together in the same habitat. Okay, so you have a group, or we call it population. All right. Ecology is a branch of science, okay, which studies the interaction between organism and the environment. So this ecology is one part of biology, actually. Okay, it studies the interaction between animals and plants, and also with the environment. That means the abiotic components. So in this population ecology, right? So the scientists who are what you call specialists in this field are called ecologists. Okay, so ecologists are scientists or biologists who are actually, their field of study is ecology. So what they have to do is they have to go into the ecosystem and take samples and study the animal behavior there, and also the animal population and so on, okay, and their distribution. So these ecologists, they are quite busy because they have to go to the field. The field means they go to the area and they do their study there. Okay, so let's look at this term, right? These two words. Now, population ecology, let's go back to population. Population refers to a group of organisms of the same species living together in the same area. So you will see here, let me switch off my phone first. All right. Okay, so here you will see the same, the word here is important, same species. Same species, this, right? Same species, right? Same area, that means habitat. So when talking about population, you're referring to the same species. Okay, let me switch off the mic. Okay, the phone. Right. Population size, you're talking about size. Size means how big. How big, that means I'm asking for the number. In that area there, in that habitat, how many elephants are staying there? Okay, if I ask for population size, I'm asking for how many animals are there, or how many individuals. If I'm asking for plants, I'm asking for, let's say, in that field, what is the population size of the Mimosa pudica? Mimosa pudica is the plant that, you know, when you touch it, it closes, the leaf closes. So I ask, if I'm asking for population size, your answer must be in terms of number. How many plants? So let's say 50 plants. Okay, in that field there, I can find 50 Mimosa pudica. That is called the population size. Size means how many individuals. Got how many animals or how many plants staying there. Okay. Next one, density. Density refers to how close are they staying to each other. Okay, density in Huayu, we say in Mandarin, we say "mi tu." That means how close are they? How do you get the density? You learned in Form 1 before. How to get density? Density is the mass divided by volume, right? So actually, in other terms of density, you take the number, you divide by the area. So for density, you must have this one per unit area. You take the number, you must divide by per unit area or volume, all right? But usually we talk about area because we're talking about 2 dimension. Okay, so density, your answer must be in number per unit area, usually meter square, okay, because we use the SI system. How many animals per meter square, or how many plants per meter square. All right, okay. So a population abundance refer to these two. That means population size and also density. So these two also included when you talk about abundance. Okay, so the population ecology, what you need to study here? You're going to study how many animals stay in that area or how many plants are found in that area, the size, we're talking about the size, and also what are the factors that affect the size. Why this area so few of these animals? Why that area got more animals? What is the factor affecting it? Maybe here got more predation, got more animals eating this one, or maybe the soil conditions are not good for this plant. Okay, maybe the other area the soil is better, or there's more sunlight. So these are the factors which affect the distribution of the population. Why here more? Why that one less? It's because of usually environmental factor, and also usually because of maybe the predators. Okay, so these are the things that the ecologists study when they study about population ecology. So what are the things? Why this animal here so few, over there so many? What are the factors affecting it? Okay, so usually what affecting it are actually the biotic and abiotic factors. So there are two, right? Abiotic, usually before abiotic are those factors which are not living. That means the environment. For example, the temperature. Maybe the temperature go there, but the temperature is better. That's why the same animal over there very few, but over here a lot. Okay, temperature, pH, right? Whether the soil, this is mostly for the plants because they live in the soil, or they depend on the pH of the soil. For pH of the for animals, it doesn't affect that much, right? Unless it's water supply. Okay, pH, then water and sources of food, whether the area has got source for food for them. Okay, then also biotic factor, right? Another factor which affects the population is biotic, whether you have competition. Maybe the area so few because they fight a lot. Why they fight a lot? Because the area is very small, so there is a lot of competition. Maybe over here the habitat is wider, is bigger, so they can space themselves out. Okay, and then they don't fight so much. Competition is less. Or maybe there's parasitism. Okay, maybe there's predation. There's a natural enemy eating the enemy here. So these factors affect why here got more, why over there got less, and so on. Okay. Other factors such as the birth rate, right? Whether how fast they are reproducing, death rate, how fast they are dying, right? Maybe because of disease. In that area, there is an epidemic. There is some viral infection. Okay. So the population they get to die off because there is penyakit over there. Emigration, that means that means they move out of the area. They move out usually because of certain reason. They cannot find food, or maybe there's a lot of competition. So they migrate. They migrate out. It's called emigration. Okay? And migrate inside, then they come in. They come in is called immigration. They come in maybe because the other area is not so good, not so much food, but now they come into this area, it's called immigration. Okay, so you can see emigration means going out. All right, immigration means coming in. So hijrah keluar is emigration, but hijrah masuk is immigration. Okay, so now let's look at before we go on, first I've got to show you this diagram right now. So here we're talking about population. Let's say this is ecosystem X. You have two types of animals and one type of plant here. Okay, so this is population. Population A, right? This is population A. Got how many of these animal here? Let's say maybe one. Here one, here one, here one, here one, here one. Okay, this is population A. This is population B. So you have one here, one here, one here. So we're talking about different the number of animals in the same ecosystem. Right? Maybe plant C, you have more, of course you should have more plants, right? So this is an ecosystem, and this is population A, population C, population B. So when we talk about population density, we want to see in that area how many individual per meter square. When you just want the population size, we just want to say in that area got how many animals. Okay, so we're going to do some counting, right? But of course, we must do work smart. We cannot be counting every plant there, isn't it? Let's say you have a very big field. You cannot be going to the area and trying to count how many pokok semalu. So we normally do a sampling. Okay, after we'll see how we do sampling. Let me check your comments. Anyone has got any question? Okay, nothing so much so far. Okay, good morning, everyone. Okay, Eileen, good morning. So let's look at factors which affect the population distribution. Why this plant, this for plants, population of plants, distribution of plants. Why the plants here in this area so much, but in the other area is so little. Okay, so these are the factors that affect. First of all, temperature. Right? Temperature is important because it affects the workings of the enzyme, how the enzyme function. So whenever a question asks you why temperature makes an impact or affects the population or affects the physiology of certain things, always talk about enzyme. Don't forget this word, enzyme. It ties in very closely with enzyme. Why certain animals cannot stay in areas which are very cold is because the enzymes are inactive. So when it's low temperature, don't say enzymes die. I repeat here again. So when you say the condition is very cold or temperature is low, you don't say enzyme denature. You don't say enzyme destroy also. But what you can say is enzymes are inactive because they are slow. They move very slowly. The function, what you call the physiology, all right? The working, how it functions, it functions very slowly. The what you call the lock and key hypothesis, where they lock into the substrate. Now remember the lock and key. So it doesn't the collision rate between the substrate and the molecule, substrate and the enzyme is very slow because they move very slowly in cold temperature. Okay, so but when it's high temperature, when something is warm, the place is warm, I don't grow well because the enzymes function very well. But if it's too high, you find the enzyme denature above 50 to 60 degrees Celsius, and the biochemical processes will slow down because they don't have the enzymes to help it go faster. Okay, for example, what is the effect? What's the effect? They will stunt the growth. The plants do not grow very well. Okay, because when it's very hot, the rate of water evaporation will be very high, so the plant may dry out. Okay, and the rate of this will be less because less water. Because photosynthesis depends on amount of carbon dioxide and also water supply. Okay, so that is temperature. Now, water is definitely very important. It is a medium of biochemical reactions in cells. So you need it time to work. You also need for photosynthesis, for transportation of minerals inside the xylem and the phloem. Okay, it also support water gives the turgidity. So areas which are very dry, all right, dry desert, you do not have a lot of plants growing there. So the distribution of population distribution very low in desert. Tundra is this area which are in the Arctic, in the north, which are very cold. Northern parts of Russia, northern parts of Canada, they are very cold. They're called tundra. Okay, very near to the North Pole. They also have very low population because number one, the temperature is very low, right? You also have less water supply because it's all frozen. Okay, it's in form of ice. It's not in the form of liquid. So when water is not in liquid, it does not dissolve. It cannot dissolve the minerals and so on. It doesn't. Your biochemical processes do not work because the water is frozen form. Okay, and that's why you have low density of plants. Okay, light is important because of photosynthesis. Okay, tie in with what you already know. You know that light is important because the green plants need it to make food. Okay, so when you have little light, that means you have very low rate of photosynthesis. So they do not have enough starch. They do not have enough food. They cannot grow very much. So when they cannot grow, they also cannot reproduce very much. Okay, pH of the soil. So this is important for plants only. For animals, we don't really have much influence on the soil for the plant or for the animal. So it's important. Some plants prefer more acidic environment. Some prefer a bit more alkaline. For bacteria, they prefer more alkaline condition. For yeast, they prefer more acidic condition. And then if the soil is too acidic or too alkaline, it's also no good because it affects their rate of absorption of the minerals. Okay. If my voice is breaking up, just right? So far, I think they're okay. I told my daughter not to use the computer. All right. So I should be all right. Just let me know in case any problem. Just put a comment there. I'll check. Okay, another one is mineral salt content. So the salt content affects absorption of water. Yeah, because of the hypotonic. So if it's too salt and too much, the water too salty, it becomes hyper. Water from the cell will diffuse out, you'll find dehydration. So your water content, the minerals content in the water must be the correct amount. Cannot be too salty and it cannot be too dilute because of the osmosis happen. Okay, water either come in or water will go out. Okay, the rest you can just read through. Now for animals, all right, what is the effect of these factors, abiotic factors? So temperature, as usual, see when it's too hot, right, temperature, animals will always go for shelter. They'll find a place to run away with the hot sun. Okay, so high temperature usually will have very low concentration of plants. For example, worms in the soil. If it's too high temperature, they'll always go for the burrow into the ground, away from the source of light and also temperature, because it's going to be too hot, right? And water will evaporate if it's too hot, then it will dry up. Okay, water, right? When there's enough water supply, definitely there'll be more animals there. So you can see around the lakes and all that, many more animals will stay with water supply when there's water supply. Breeding site. When there's a breeding site, for example, a place where they can build their nests, they have a cave, so you have animals staying there because they have a place for them to breed, or a place for them to bring up their offspring. Okay, there's also food supply. When there's food supply, this is very important, right? If you can't have enough food supply, the animals cannot stay, cannot live there. So when there is food, when there is prey, there will be predator there, staying there at the same place. Another answer would be probably mates. Yeah, they have mates. So they have female and male, right? They will always stay together because they have the need to reproduce. Okay, so these are the population distribution, the factors which affect the animals. Just now for the mates is actually biotic factor. So we're talking about abiotic factor here, is it? Yeah, okay, abiotic factor. So let's look at the population distribution. Distribution means the pattern. How near are them to each other? How near are the animals in that habitat? Distribution, okay, is how much are they spread out? It's called distribution, spreading out. So if it's distribution of different patterns, so you may see you have clumped, random, or uniform. Okay, what does it mean? That means if you go to the area you want to talk about, let's say pokok semalu. I want to, let's say this is a field. I know the field has got pokok semalu. So I go into the field, I want to see how are the pokok semalu growing. Are they growing in certain clumps? Clump means got one group here, I can find many here. Then the rest over there are empty. Then I can go to another place or I find one small group here. So this is called clump. Okay? Or is it random? Random means there's no particular pattern. You have one plant here, one plant there, one plant here, one plant there, everywhere. So this is randomly distributed. No pattern. Okay. For uniform, there is generally a certain distance. Okay, certain distance from one to the other. So they are distributed all over the area. Usually there will be a certain distance. Right? Distance usually is when we plant them. We will have a certain uniform distance. But whenever you have naturally occurring environment, usually because of the seeds, right, they come and blow over and so on. So it's usually more of a random. Okay, and clumping also because that area has got more resources. Maybe more water here. Maybe over this side, this is more water. Sorry, maybe here has got more water. So that's why here you have a clump here. Maybe this soil is more wet, all right, and over the other areas is not so wet. So maybe here, over here, so you have more plants growing there. Okay, random is based on just random distribution. Maybe all the soil is equally fertile, just happens to where the seeds happen to blow there. Okay, so you have three patterns. Clump, right, which is separated groups, different groups separated in different small groups, right? There's uneven distribution. It's not even. This area got more, this area got less. Okay. Random is a free pattern. So they have little interaction because they're so far apart, right? They are randomly distributed. This one can be plants, this one can be animals. So it could be clumps, animals, one group stay together there. Okay, so you consider as animal or plants can. Uniform, usually this one is for plants. Okay, they distribute all over the area. So it's because they're competing for limited resources. Okay, so now how are we going to estimate in that field there or in that area you want to study? How are we going to find out how many plants? Okay, so first thing to do is you count, right? You go to count, right? So if your area is small, no problem, you can count all the plants, right? Let's say the field is just like the field in your garden. Let's say your garden, right? Your garden is in your house. Your garden can be very big, right? You want to find out how many pokok semalu are growing in your lawn, in your garden. You can go and count. You can just walk over the whole place and just see how many you can count right now. Right? That is your population already of your pokok semalu in your garden. But if you're talking about a bigger area, let's say the school field, right? Our school field is so big, right? You cannot be counting all the pokok semalu in your area because it is too difficult to do so. In fact, especially when there's too many. So what we do is we do a sampling. This is, we talk about ecology, this is one of the methods that we do. We do a sampling. That means the sampling is a representation. It represents the entire area. It's difficult to count every plant. It's not possible. Okay, especially let's say we want to count about ants also. If ants in a colony can come up to thousands and tens of thousands of animals in the nest, So it's not possible for you to count how many. Not possible. The whole ant hill, you cannot come up to maybe 20, 30,000 ants. It's not possible for you to count. It will take forever to count, isn't it? So you got to do sampling. So we're going to learn two methods today. One is for animals, one is for plants. How to do the sampling. Okay, so this is called sampling technique. Right? If you do it accurately, if you do it in the proper way, it gives you a correct representation. Even though you didn't count each one, but it gives you an estimation. Estimation is ramalan or jangkaan. Roughly inside there, got how many plants or how many animals. It is an estimation. So you use this sampling technique to study the populations. Okay, you want to know in that area that you study, right, a confined area, got how many, roughly right, roughly got how many plants without actually counting each one. So this is something to statistics, right? This is one of the methods in statistics. We do sampling. Sampling, if done correctly, it will give you a very correct, quite accurate reading. That means quite near to the actual figure. It cannot be 100% actual figure, but it will be very near if you do it correctly. Okay, let's see how you do it correctly. So you have to do sampling. Sampling means to take samples. Take samples. Take sample means you have one example, this whole area. I cannot be counting all. I count a few only. That's called sampling. I take from this area a few, from that area a few. I count, all right, and then I will do estimation. Okay, so a sampling technique involves you have to collect sample, you have to count, and keep observation on the organ. So this is plants and plants. Later on, we go to plant. Okay, after that, go to animal. So there are two different techniques. Okay, sampling technique. Number one is for plants, we use this one, quadrat sampling technique. For animals, we use, here's we got. Okay, sampling technique, first we use the Capture-Mark-Release-Recapture, CMRR for short. So for example, please get the full term. You can put CMRR there. Okay, but make sure you first time whenever you do write the full term first time at least once. Okay, so for animals, you use CMRR technique. This is a sampling technique. For number two, second one is for plants. Plants, you use quadrat sampling. Okay, so now you understand what are the two methods. First are two techniques. For plant, you must use quadrat sampling. For animals, you use CMRR. Okay, why? Because animals can move. Animals are not there all the time. Okay, if you do sampling, you put a quadrat there. Quadrat is like a frame. You put a frame there, and then you go to the field and then you count. But because if animals can move, once you see the frame already go animal, the moment you want to count, the animal jump out already or the animal move already. So it's not suitable to use quadrat sampling for animals because animals can move. But for plants, they don't move, right? You put the quadrat there, you see the plant there, the plant is there. It doesn't move away. So that's why it's more suitable for you to use quadrat sampling for plants, not for animals. Okay, now let's look at the method. Okay, so far okay lah, everyone? Let me check, am I going too fast? Nothing, so far. Just put in the comment there, anything, right? I know you can't talk to me, right? But I will be checking your comments from time to time. So just let me know, right? Afterwards, we go to Google Meet, right? We can have a two-way interaction, right? Now let's look at quadrat sampling technique. It is used for what? For distribution of plant. You want to see the pattern of distribution or population of animals which are inactive. That means they don't move really very fast, like snail, right? Some animals that don't really move. Ants cannot, ants they move quite fast, Once you put the quadrat there, they'll run away already. Okay, so usually I do not recommend for animals, but the textbook says you can use for animals. Here we should move slowly, so I put it in, right? So we also follow the textbook, but basically usually it's just for plant. To be on the safe side is plants. You use quadrat sampling for plants. For animals, you use the other one, which is called CMRR. Okay, let's look at how do we make a quadrat? What is a quadrat? A quadrat is just a frame. Your frame can be square, it can be rectangle, it even can be round. Okay, it can be a circle, as long as you know the area. So you have to prepare a quadrat first. Now, how big? So how to make quadrat? You can make from wood. Okay, you take 4 pieces of kayu, right? You just paku them together. You just make a frame, right? And then your frame square, right? Then you find the area first. So you can use wood, you can use string, you can use rope, or you can use metal, as long as it's a frame. That's all. It can be metal, it can be kayu, right? You can make a frame. And in between, let's say you have a 1 meter, 1 meter, your 1 meter is actually quite long, it's about your arm span, meter. You make one frame like that. Then in the middle, you can put some strings to make even smaller squares. It's up to you. You don't have, also never mind. You don't need a small squares inside. You don't really need. You only need the outside frame. Okay, so it can be subdivided. You don't want to subdivide, also can. You want to subdivide even better, because you can. This subdivision is usually for coverage. How many percent is covered? That one is more for coverage area. Okay, so let's say now this is the field that you want to. This is a field. All right, I want to check on this plant, flowering plant. Don't know what plant. Okay. So what I do is I go to the field, I put it on the ground. This one, if you do it randomly, I don't want to purposely choose the area I want to get sample. I just put it wherever I want. That's called random. Random, I just put there. If got, it's got lah. If don't have, don't mind, doesn't matter. So in that, let's say I put the first quadrat, I put it on the ground. So happen here, I see how many? 1, 2, 3. Now this one no need to count is if it's less than half. If it's half or more, you have to count. Okay, so here I will assume it's 4 right? If you have more than half or half, you actually add inside. So you just put here the first quadrat number 1, you get how many? 1, 2, 3, 4, let's say. Then I take my same quadrat, I put another area. Let's say I put it here. The second time, right? I put it here, and I get how many plants? I get 1, 2, 3, 4, 5. Okay, the second quadrat I get 5. All right, you write down, jot down in your notes. Then I go and take the same frame. I walk somewhere else. I put it on the ground. Let's say I go to here. Randomly, okay, don't want to be affected by, I purposely want, I purposely put that. Now there a lot, I purposely put that. No, it's not. Once you do it this way, it's affected by your own judgment. Your result is not accurate. You just either do it randomly or you do it systematically. So there are 2 methods for you to put your frame. Okay, afterwards I show you where. 2 methods. So the first method I'm talking about is random. Random means you just simply put your frame anywhere you want. Okay, don't care whether it got or don't have, also don't care. Don't want to be purposely, I want to plan, or I must put at a place where I can see the plant. No, just put there. It's called random. So like you see here, the third one, I only got 1. So you jot down. Third quadrat, 1. Then the fourth quadrat, I walk somewhere else. Then I put my quadrat here. Here, so happen nothing, right? The quadrat number 4, 0. Okay, understand? So you walk around the whole area, you bringing your quadrat and putting it on the ground, and you just jot down how many. Now, how many quadrat to take, how many sampling you must take? Do I take 5? Do I take 10? Do I take 20? Okay, it's up to you. All right, it's up to you. You know why? Because you put your reading in the answer. Doesn't matter how many times you do your quadrat, you will have an estimation. Now, one thing to remember is the more sampling you take, that means you do 20, 30, of course, take more time, right? Your answer will be more accurate. Okay, remember, this is statistics. The more sample you take, the more accurate your answer will be, actual answer. So if you do only 5, let's say you're very lazy, no time, lazy, so it's hot now, under hot sun, I don't want to walk so much. I do 5 only. I just put 5 only, I get 5, and then your answer will be, of course you get answer, but your answer will not be as accurate as the actual answer. That means actually how many plants are there? If I do only 5, I get the answer maybe very far away from the real answer. But if I do 20, 30, 40, if I do more sampling, my answer that I get after calculation will be very near to the actual number of plants there. Okay, so this is something for you to remember. This is an assumption. When you want to increase your accuracy of this method, always increase your size of sampling. That means you do more times. The quadrat you take more times, it will be accurate and more better answer. Okay, so let's say you've done 5. Okay, the 5, there's a 5th one. I go and go back. I go to this area. All right, you don't go back to the same area, of course. You want to go to do your sampling different area. This is called random. Now, what is the second method? Is called systematic. There's another method you can do it systematically. Systematically sampling. We call it systematic sampling. What happens is this is your field. All right, this is your field that you want to investigate. All right. Now, what I do systematic is I make sure I put it at different distances. So my first quadrat I put here. Okay. Then I measure a distance of maybe 1 meter. Then I do the second quadrat here. Then I do the third quadrat here. That's systematic. Then after that, the next one I measure again 1 meter. This one. So you go like this. This is called systematic sampling. So it also works. All right. Either you take random or you do systematic. So here you have 9 quadrat. If I were to put it in a systematic way, either way you will still get your answer. But do not do like what I said just now. You purposely want to think, oh, I see they got a lot. I purposely want to count. Then your answer will be very high. Your answer, your estimation will become very high because you purposely take the place where you got. Okay. And then I purposely, or I lazy to count. Let's say I lazy to count. I purposely put that one don't have. I purposely put there. Then in the end, your answer is going to be very little. Okay, your answer of the number will be very little. So don't get it affected. Don't make yourself affected by where you want to put it. Either you choose random sampling or systematic sampling. Okay, your answer will be more what they call accurate to the actual answer. Okay, so let's get this. Let's do this question now. So now this area, importantly, it doesn't have to be 1 meter by 1 meter. It can be 0.5 meter times 0.5 meter. Okay, it can be. It means you have a small quadrat. Doesn't matter. Now, why smaller quadrat? Maybe your plant that you want to measure is a small plant. You don't need to bring a quadrat into the field. Your plant is very small one, so you can put 0.5 by 0.5. Okay, you can consider that. But you make sure it's consistent. Every time you do also use the same quadrat, right? So depends what's the size of the quadrat, depends on the size of your organism to be studied. So if your tree is very big, you're looking for a big tree, you cannot be using such a small quadrat, all right? You cannot put on the tree also. So if you want to do a quadrat sampling for, let's say, a big tree in the forest, maybe you need to have 10 meter by 10 meter. But how to make such a big quadrat? Actually, you don't have to make. What you can do is go to the field or go to the forest. You put pakukan one place here. You take a tali raffia. You measure 10 meter, and then you measure another 10 meter. That means on the spot you make a quadrat with tali, with string. You can make a quadrat there. Okay, you can make a quadrat. You don't have to bring 10 meters. Is quite difficult to bring. 10 meter is very big. You cannot bring the frame. It's too big. So 1 meter by 1 meter you still can do. But 10 meter by 10 meter you cannot. You have to go to the forest. You make your quadrat there on the spot using string. Okay, so you paku here, one place here. Then here 10 meters you measure. Another place 10 meters, 10 meters, 10 meters. Then you have a makeshift quadrat, then you can count the number of plants that you want there. Usually trees are. Okay, tree cannot be 1 meter by meter frame quadrat. Okay, so for small epiphytic plants like mosses and lichen, maybe 10 by 10 cm also cannot. 10 by 10 is very small only. Okay, 10 by 10. Now this is 10 cm, right? 10 cm is just like this only. So you make a small quadrat, right? Then you can put it on the mosses and the lichen on the rocks, on the tree bark and so on. You can even use a transparent plastic sheet. You draw the quadrat there, you put it on the tree bark there, and you can see, okay, inside the box there how many plants, and you count. Okay, or for herbaceous plants and grasses, using 1 by 1 meter. Circle also can, but make sure you know the area, how many meters square. Circle is more difficult. You have to make use of diameter and pi, right? You have to use a pi r square to find out the area. So usually square is easier for us to handle when we talk about finding the area. Doesn't matter, you can use circle. Okay, for plant creepers and shrubs in the forest or in the whatever, you 5 by 5 meters. That one 5 by 5 meters also you cannot make the quadrat. You have to go to the field and you make it there using tali, okay, string. For trees, trees are definitely 10 meter by 10 meter. That means you really have to go to the forest really, this one. Okay, so quadrat can be thrown randomly. That's one random. Another one is systematic. All right, so I mentioned just now. You have to do at least 10 quadrat in order to be accurate. I mentioned just now, the more number of quadrats you take, that means the sampling is more or more quadrats, you will have a more accurate result. But usually 5 also can. So for calculation here, let's do 5 only. All right. Now let's say there are 3 ways you can find the distribution using this quadrat metric. Either they ask you for density, how close are the plants, or they ask you to find the frequency, that means how often the plant is there, found in the place. Another one is percentage coverage. Percentage coverage is usually for mosses, for trees which have a lot of foliage, a lot of canopy. So you want to see how much of the leaves are covering the area. Okay, let's look at one by one. Now, density. How to get an answer for density. So here, the question asks you, what is the density of the plant that you are studying there? How close are the plants in your field, right? So what you do is you look at the formula. You gotta remember a bit this formula, you gotta remember. So try not to rely on the question give you formula. So it may not give you formula. So you got to try to remember the formula. First thing to do is you count the total number of individual species studied in all quadrat. You see in all your quadrat, let's say 5 here. Now you do 5, is it? Quadrat number 1, quadrat number 2, quadrat number 3, quadrat number 4, quadrat number 5. That means you do 5 sampling. You throw it 5 times. You throw your quadrat 5 times. And then the first time, the first quadrat, you find about 5 plants inside there. So you jot down 5. The second quadrat, you throw somewhere else, right? Random sampling, you got 4. So the next one got 4 plants. Okay, you can count 1, 2, 3, 4, right? The third time got 3 plants inside there. Now the fourth time you throw, don't have the plant that I want. The plant that's pokok semalu, I don't see, but I see others. Others don't need to count. You only count your pokok semalu, right? Don't have so kosong. Then the 5th time you throw, you get 3. So what you need to do is, on top here, this number, this formula, you add up all this. You add up all this. How many individuals have you found in all your quadrat? Add up for all. So you have your 5 plus 4. Okay, please jot down. Write down the formula, do some questions. 5 plus 4 plus 3 plus 0 plus 3. All right, this is what is the all quadrats. Then divided by the total number of quadrat used. How many quadrats you use here? You use 1, 2, 3, 4, 5. Okay, so 5 quadrats. And then what is the area? Every quadrat, each quadrat, 1 quadrat, what is the area? The area usually, is given here in the question, 1 meter square. So you have to times 1 meter square. Okay, so your answer for density is always per unit area. That means per meter square. For answer for density, the unit for density is not just how many plants, it is how many plants per unit area. So let's do some calculation. Okay. 5 plus 4 is 9. 9 plus 3 is 12. 12 plus 3 is 15. Okay, correct? 5, this is 6, yeah. 15. Divided by 5 times 1. 5 times 1 is 5. So your answer will be 15 divided by 5. You get 3. Now, what's the unit? Here on top, number of unit, but at the bottom, you see per minute, per unit. That means it is how many plants per meter square. That means in 1 meter square, that means purata, or we call on average, in 1 meter square, you will get 3 plants. This is how dense they are. So if the number is higher, means that there are more plants in the area. That means more dense. A lot means a bit cramped, we can say. If the plants there, let's say 12 plants per meter square, that means in 1 meter square, you have 12 plants. If 3 plants per meter square, that means in 1 meter square, you have 3 plants. That is a bit spacious, right? Not so near each other. So this is how you use the formula. Okay, this is for density. Density, make sure is per meter square. How many individual per meter square? Or you can put negative here. Plants meter negative also can, right? Or individual. You don't have to say plant, you can put individual. So how many plants? Now this one can be a decimal. This number can be decimal because it is per unit area. This number, let's say 3.2 is perfectly okay. Let's say you get answer 3.2. You can leave your answer in decimal because this is density. How many point something per meter square? Okay, you don't need to round up. Okay, all right. Now for the frequency, all right. Frequency. How often when you do your what you call the quadrat, how often do you get your plant there? What is the frequency? How often can you find the plant there? So here you see, let's say first quadrat, you get 2. Second quadrat, you get 3. Third quadrat, you get 1. Fourth quadrat, you get 0. Don't see a plant there. And fifth quadrat, you get 2. Now, what is the percentage frequency? So percentage is like this. You count the number of quadrat which contain the species. So you can see here, the first quadrat I got. Don't add up number. Don't add a number. You just take got. The first quadrat got. The second quadrat also got. The third quadrat also got. The fourth quadrat don't have. I don't get any plant. All right. The fifth quadrat other. So now I can find that it's only 4. 4 of the quadrats other. 4 of the quadrats have the plant that I want to study. And then divided by how many quadrats did I use? Or how many times did I do the sampling? I have 5, isn't it? Out of 5 times I throw, only 4 times I get the plant there. So it's 4 divided by 5. Okay, then you find the frequency is usually a percentage. So times 100%. Okay, so this 4 is not adding up. You don't add your 2 plus 3 plus 1 plus 0 plus 2. Not like the first one. You just count how many quadrat got this one. So it's 4 other. And I do 5 times. So 4 over 5 times 100. So this is 80%. Okay, so frequency is 80%. Okay, this one don't count. Don't add up your 2, 3, 1, 0, 2. Don't add up. Okay, it's just how many quadrat other plant that you want to study. Okay, so second formula. Now, third one. Now, this is percentage coverage. Usually for coverage for like moss, lumut, all right? Because like us also, yes, because sometimes, sorry, the leaves are very widespread. It covers an area. So it's sometimes for plants that are difficult to identify. This is 1 pokok, this is 2 pokok, this is third one, because they're all clustered together. You can't identify this is how many plant, how many plant, how many plant. It's all covered like moss. You cannot count. This is 1 plant, this is 2 plant, 3 plant, because it's all very small and it's all green. How to count the plant? You cannot count. So you count the coverage. How much area is covered by your lumut? Okay, by your whatever you call that, the moss. All right. So coverage, it can be for plant in terms of the leaf coverage, or it can also be this one, lumut. All right, let's say lumut. Let's say you put your plastic, your quadrat there onto the rock there. You can see now this one, roughly 0.3. That means 1 One whole area is 1 meter square, all right? 0.3 is about 30%. Only 30% is covered. So you get a rough estimation. For this one is roughly or almost 50%, right? 0.5. Then this one 0.8 now. Here almost done, right? Almost covered. So you get an estimation of each quadrat first. This is the coverage area. So how do you do your formula? You add up all the area that is covered in the quadrat. So what you do here is percentage coverage. You add 0.3, 0.5, 0.8, 0.4, 0.5. Okay, that is your area covered by all the quadrats. Right? How many quadrats you use? If you have 10, you carry on, you add on some more to 10. Divide by, okay, divide by what? Divide by the total number of quadrats. It's always at the bottom the same. What is total? You got use 5. So 5. And don't forget 1. Each one quadrat, what is the area? Is 1 meter square. Okay, you can see 1 times 1, isn't it? Each quadrat is 1 times 1 meter, so 1 meter square. Here, you got to do a quick calculation. So 0.5, 0.8, 0.8, 1.6, 1.6 is 2.5. Okay, 2.5 divided by 5, correct? If I'm right, 0.8, 1.6, 2, yeah, 2.5. So now you have your calculation. It's going to be 2.5 divided by 5. So I get the wrong calculation. 2.5 divided by 5, you get 0.5, right? It should be 0.5. Okay, this one, look at the, if as a meter square, the bottom here, that means per meter square. This is per meter square. 0.5. Oh, this is also 0 meter square. So your percentage coverage is, oh sorry, forgotten, times 100. Times 100 because it's percentage. So it's going to be this one, sorry, times 100. Forgotten 100. It's going to be percentage. Yeah. Answer is in percentage because percentage of coverage. Your meter already cancelled out already. Your meter already cancelled out. No more meter there. So your answer will be 50%. Understand? Okay. So remember it's percentage coverage. So you have to times percent. And your answer is always in percent because the top is meter square. The bottom is meter square. Actually, the meter already cancelled out. So the answer is in percentage. Okay. So this is sampling technique for plants. We use quadrat sampling. Quadrat is a free money, right? It can be square, it can be round, whatever, but you need to remember the formula how to get it, right? Okay, so now let's look at animals. How are we going to do for animals? Animals, you can't use quadrat. For example, you put your quadrat there, right? You throw your quadrat there. By the time you walk over there, let's say the quadrat kena the plant, the animal inside there got 3 animals. By the time you walk over there, the animal already come out or walk away already. So how to say, I don't have, you cannot see, isn't it? Already gone off. So you do not use it for animals. You use it another method. This is called CMRR for short. Capture, mark, recapture, release, and then recapture. The word is very simple. First of all, you need to capture. That means you got to set a trap. You capture. Capture how many animals that go into your trap. Don't care, right? Okay, how many you can. So this is how much you capture. Okay, wait. Let's go into this one first. Let's estimate the population of size of animals such as foxes, sharks. Sharks are very difficult. How are you going to set a trap for sharks? Usually for small animals, snails, millipedes, okay, insects, they can move in an area, all right. Maybe wood lice, okay, under a tree, kutu, whatever, ants also can. So ants is quite common use for this one. Okay, initially what you do, you capture first. You put a trap there, you capture, let's say, give you an example. This is the area under study. You put your trap there, all right. How many animals trapped there? This is your first capture. You set a trap, right? This is the first capture. The first capture, let's say you get 80 animals, 80 ants. First capture, all right? Now, once you capture it, you have to mark it. You have to mark it first. You have to mark it with identification. That's something that you can recognize. You put an ink, a color of ink that cannot wash out by the water. You must put waterproof ink. And it must be not toxic. It cannot be harmful to the animal. It cannot be poison to the animal. Of course, you put it there and then it's poison, of course the animal will die, isn't it? Then the second time you want to catch it, it's already gone. You cannot catch it again. So you have to make a ring or a tag, something that you can recognize. You put a dot, you put a marker pen, which is not hard, the ink, make sure it's not hazardous. It doesn't don't have a chemical, don't have the benzene or the xylene. Make sure it's not harmful to the animal. Or you can use color ink. One not common example is the Indian ink, all right, or cellulose paint, so that they cannot be washed by the rain. Now the syarat here is must not injure the animal. It's not poisonous to the animal, the ink. It must not stop their movement. Let's say you tag something so big until they cannot go back into the hiding place. You go tag the ear there, whatever. So big, the whole thing, so big until it's so heavy, the thing also cannot walk, all right? It cannot hinder the movement, and also cannot make them so easily to be seen by the enemy or the predator. You cannot endanger their lives. For example, the animal is black color, right? You're going to put one big red thing there and then you easily to be seen by the predator of the animal. Then the animal will go and the predator will eat up. Then later on it affects your study because these animals has already died, eaten by the predator. So you must not harm them. Cannot put something that is so conspicuous, so red dot there, yellow dot, until the animals get easily eaten by another animal. Okay, so syarat. It cannot affect, it cannot harm the animal. Okay, then after what you do after you mark all of it, let's say the first time I capture 80, I mark all 80 with the same method. All right, I mark all 80, then I release. You release it, let it back, go back and mingle with your friends. Mingle with those who have not been marked. Let mingle, don't care, right? Let them say bye-bye, all right? Let them go and mingle with their friends, with their family members, okay? Then after that, allow them to mix. Make sure got enough time. You don't go and let them go and then go back and then come and capture again the next hour, because not enough time for them to mingle. You want to have a fair sampling. You want your statistics to be more accurate. You want your experiment to be accurate. So you must let nature do its course, right? You must let enough time. Usually we leave about 2 days, 2 to 3 days. And don't leave too long also, because if you leave it too long, some of them could have died, all right? Maybe they could have had new babies, so the population will change, or they would have other animals come into the area, you have emigration or immigration. So other factors will affect the population if you leave it for too long. So usually you want to catch again the second time, you don't leave it more than 1 week. Usually we take it about 2 to 3 days will be the best, or even 4 days maximum. So after a few days to a week, maximum a week, then you do another recapture. Take the same trap, put the same place, and catch again. Okay, so now catch again is called the second capture. So remember, first time you already captured first time. Then you mark all the ends. You mark it all, right? Correct with the suitable marking. Then after that, you release. Make sure you release it and leave it enough time for them to mingle with their friends and family members. All right. After that, you set the trap again. You capture again second time. This is called the second capture. Now, second capture, you will find that it could be more than 80, it could be less than 80. It doesn't matter. But you will find that inside this sample, you will have some which are already marked. That means they are second time unlucky. First time capture is the same animal. The second time also capture the same animal because you already got marked, isn't it? And also you will have find some which are unmarked, because it means the first time you didn't capture them. But now because mingle, right? So now the second time you put the trap, you get this unmarked one. So you have a mixture of marked and unmarked, isn't it? All right. So doesn't matter. Don't care whether it's more than 80 or less than 80. Doesn't matter. It's not going to affect. But you make sure, of course, definitely you have marked and unmarked. Okay. So you have these two groups of animals in the same trap now. This is second capture. All right. So second capture, you have to count. Now, how many are marked? Let's say second time you capture, now you have got more. Let's say now you got 100 second capture. But out of the 100, maybe you find let's say you find 65 is marked, and another 35 is unmarked. Okay, so 65 is marked, let's say lah. Now you got this number. You have got 80, you have got 100, you have got 65. Now look at these three numbers. This one unmarked, don't count. Don't need already. You throw it away. Don't need to look at it. You want to see the first time you mark how much, how many. The first time you capture how many. The second time you capture total is how many, and the third time is the second time capture how many are marked. Just need these three numbers only. You put into a formula, then you can find your estimated population. Okay, how many ants are there roughly lah in your habitat, in the area that you want to study. Okay, let's look at the formula. So now you see the formula you use is the number population size is A times B divided by C. So always I put it easy this way. A times B divided by C. What is your A? A is the first time you capture. Altogether capture how many? Then times B. B is the second capture. Second capture is 100. Just now the first capture 80. Second capture 100. So this is your A. Right? This is your A. Let me write down the black color. Okay. This is your A. This will be your B. And divided by C. Now C is how many they are marked. Don't take the unmarked. Sometimes the question want to test your, K-bar bit. They give you the unmarked. So make sure you don't use the unmarked. We don't need this. We want to find the marked. So how to find the unmarked? The unmarked will give you. If I give you 35, I just take 100 and minus 35, I will get my 65. Correct, not? Okay. So make sure it's marked. Put inside the formula, you should get your estimation. Now this number must be a whole number. It cannot be a percentage. It also cannot be a decimal. Because the number of animals cannot be point something. How can you have a 0.5 animal? Okay, you cannot have a 0.2 animal. So this number must be a full number. So let's put in the number 100. First time 100, right? No, sorry. First time 80. First time 80. Second time 100. Divide by 65. So you punch in your calculator. Alright, you punch in the calculator 80 times 100 divided by 65. You will get how much? Enter. Okay, now 1, 2, 3. So usually 1, 2, 3.7. Now you don't put like that. Now you just put 1, 2, 3 individuals. You don't have a decimal. So you don't put decimal there. You can't have point something of an animal. So you just 1, 2, 3 animals. So correct not? I think 80. 80 times 100 is the second capture, equals to 8,000 divided by 65 the second time. 65. Sorry, 65. Yeah, okay, now 8,000 divided by 65. You will get 1, 2, 3. Okay, that means in your habitat there or in your area of study there should be 125 as 123 individuals, roughly estimation, because you didn't count all, not possible you count also. Okay, so that is your answer. So this one, let's look at this example. Look at this. This is question. Now wood lice. We caught 120 wood lice at random under the tree. So this is considered the first capture. So put it as A. Okay, and mark each one of them using a dot of cellulose paint. The wood lice are then released. Okay, so 120 are released. After 2 days, he made a recapture, and then the recapture 100 are caught. So this is the second recapture. This is the second capture is called B. Among them, 24 are found to be marked. So this is C. So what you need to do is 120 times 100 divided by 24. Okay, so this is 120 times 100 equals divided by 24. So your answer will be 500 individuals, or 500 individuals, animals. Okay, so that is your answer for. So remember your formula, A times B divided by C. Okay, but you must know what's ABC. Okay, lastly, what are the assumptions? This is a popular question. When you do your CMRR method, what is the assumption that you make? in order for it to work, in order for you to make sure that you get what do you call this is estimation of your sampling. So you must assume that when you're doing the investigation, there is no predator in the area. That means they don't die because of what animal eat them. In fact, when you talk about doing this example, this study, within that few days they do the experiment, between the first capture and the second capture, you must assume that the population is stable. That means the number of animals there do not become less because of dying, and they do not increase because of new birth. So you must assume that the population is static or stable or consistent. Constant. That means you during the study, first capture, second capture, you must not assume anyone have died or any animal has born. So you consider it's no predator, they are not harmed, they didn't die. So you consider the population is stable, no changes during your first capture and your second capture. Okay, so example, you must assume that the immigration or emigration do not take place. That means no new animal come in or no animal go out. Or you can also assume that the rate of immigration is the same as rate of emigration. How many go in is equal to how many have gone out. So you consider it is an assumption. We do not know whether it's true or not, but we assume that the population is static. Okay, and then new animals, the rate of death is equal as the rate of birth. How many are born equal to how many have died. Okay, we call it death, or rate of death is called mortality rate. How many new animals born within the first capture and second capture equal to how many animals that have died within the same time period. Okay, and you must make sure that they have mixed randomly. That's why you cannot immediately let go and immediately go and catch back. You must give a few days at least for them to mix randomly. Then your result is more accurate. Okay, so in order to make sure that it's even more accurate, remember I told you, you go and do large sample. That means you take more times. You don't trap. You don't make one trap. You go and put 10 traps. Put 10 traps, and then when the second capture, also put back the 10 traps. So that means you got more sampling. Same thing for your quadrat. You don't throw 10 times. You throw 20 times, 30 times. Your answer will be more accurate. And the animals are captured at random. Just let them fall into the trap yourself. Okay, don't need to purposely make them go into the trap. Or I want to find this animal, I want this animal, I purposely go and trap the thing. No, just let it randomly happen by itself. Okay, so these are the assumptions which will come out in objective questions. It can also come out in the structure. They give you what are the assumptions that you use when you carry out the CMRR method, okay, or CMRR technique. Okay, so I have done this, right? So afterwards, we go to Google Meet, and then we're going to do some more questions and some calculations, right? To make sure you really master this. It's not difficult. It's very easy. All right, okay. So I think we will stop here. You can refer to your textbook. It's roughly the same, except that I've got more information here. I just go through this, right? The formula is the same. For quadrat sampling, we have 3: frequency, density, and percentage coverage. For animal, they just ask you for the individual, how many only. Just remember formula A times B divided by C. Okay, like this one. And then I'll talk about one. This one can be done as the experiment in the lab. They give you kacang to represent a certain animal, and you're going to scoop it out, and you're going to do the calculation. This one can be one of the questions for Amali. Okay, the quadrat sampling. What I mean is CMRR. They give you kacang, they put in a bucket of sand or bucket or whatever, of this sawdust. You scoop out, consider your kacang is an ant. Then you count and find the answer, the population estimate. Okay, so I will see you in my next class. Okay, so whoever is there, thank you for watching. So my girls, I will go to Google Classroom. All right, okay. Thank you, girls. I'll see you afterwards. Okay, bye-bye. Bye-bye, everyone.